Welcome everyone to the scientific session here at the Fiocruz Foundation in Bahia. It is my pleasure. I am Ronald Blanton. I am a longtime collaborator with many of the people here in Bahia, but especially here at the Fiocruz, and especially with Dr. Mittermeier Hayes. I am a physician, but have done research in infectious diseases for 30 years or so, particularly in schistosomiasis. I have the pleasure of introducing one of my colleagues from the United States, Dr. Jeffrey Long. Jeff was trained initially in California, which is his home, at the University of California, Santa Barbara. He worked or got his bachelor's degree in anthropology, and then went on to do a master's at the Arizona State University in anthropology as well, and then moved to the University of Michigan to get another master's in human genetics and anthropology, and then finish his PhD there. He went through, this is the life of academics in the United States, so we're going to make a trip all around the United States for a while. He did a couple of postdocs, one in Virginia, one at University of Pittsburgh, and then went to take a faculty position at the University of New Mexico, from where he went to the NIH and was working mainly again on human genetics. He then moved to the University of Michigan as a full professor, and finally back to the University of New Mexico as a professor in biology and anthropology. His focus has been on investigations of human genetics, in particular ancient DNA that's in the human genome for other ancient hominids. And also a lot of emphasis of his work has been on the genomics of Native Americans and human variation. So it is a move that he's going to talk about to think about the genetics of schistosomiasis, which we began almost at the beginning of all of our work back around 2011 or 2012. We have an idea of where we would want to go with genetics, but in order to get it done, Jeff was very instrumental in getting us to the place that we wanted to be. He has his expertise in statistics and mathematics. That was not one of my fortes, but certainly something that he is extremely experienced and strong in. So I would like then to have Jeff come up and then give his presentation.
Well, thank you, Ron. Took me down memory lane there, and a long time back. I'd like to start out by thanking the audience for allowing me to speak in English. It's better this way, given my skill with other languages or lack thereof. And also just point out that I'm going to be talking mainly about a paper that I published with this group last year, and it was our sort of initial voyage into population genetics. As a human geneticist, I think there's really no such thing as human genetics, that there's just genetics. And you have to understand genetics to understand humans, but that also helps with other organisms. And it's always a pleasure to work with other organisms and look at things from a different perspective. Okay, so you don't need me to tell you about schistosomiasis. This is your area of expertise. I've learned a lot. It's caused by blood flukes in the genus Schistosoma. Only one of these major species is present here in Brazil, Schistosoma mansonii. It's an important cause of global mortality, and occurs in Africa and Asia, and it may account for up to 200,000 deaths annually. In the United States, we're not too aware of schistosomiasis because it doesn't generally occur in our country. But as members of the world population, it is important to us. It's of interest that schistosomiasis was actually introduced to Brazil and other portions of the Americas through the transatlantic slave trade. So that's another unfortunate consequence of that trade. That very brief background, I'd like to talk about the goal of this study that Ron brought me into somewhat over a decade ago. And basically what we'd like to do is use genetic diversity in the worms to track the spread of infections in human populations. So all of the genetics I'm talking about are genetic variations that are in the worms, and we haven't looked at any genetics of the people who are carrying these worms. But the main questions that we would like to ask and that we have asked are, do the parasites form genetic clusters that coincide with the communities of their hosts? So in other words, if I go to a local population here in Brazil or another place, do those parasites form a genetic cluster on their own? The next question that we would like to talk about is, do the parasites form genetic clusters at the sub-community level? So if we go into a community, we might find that there is a community cluster, but within that community, there might be clusters as well, maybe that reflect particular family dwellings or water sources within the community that some people use and other people don't use. And then finally, what we were interested in is, are there genetic relationships between the parasite groups in geographically distant human communities? In other words, if we look at a community, are those parasites related to the parasites of the nearest community? Are they related to parasites that are in communities far away? And I think that we found some pretty exciting and perhaps unexpected results that I'm pleased to share with you. Once again, there are many people here who know much more about the parasite than me. And in humans, the parasite is long-lived once you get an infection. I have read that it's 5 to 6 years on average. I've been told it may be much longer, decades longer. It reproduces sexually in humans and releases fertilized eggs. The adult worms have mechanisms to evade our immune response. And so that makes it tricky to control the disease. The pathology itself is not due to the adult worms, but it emerges from our immune response to having large numbers of eggs that become trapped in our tissues. And drug treatment is 100% effective in clearing infections, but there is a problem since we don't develop immunity that once your infection is cleared, you're just back to the beginning and are equally susceptible to somebody who's never had it before. So it's not like influenza or COVID or something like that, where you might at least have some immunity to a strain that you just experienced. Here's a slide showing the schistosome life cycle. I was happy to see that Lucio showed it earlier this week, so I felt like I was on the right track looking for sources. And it shows our complicated life cycle of the worm, where if we start where that circle is, there are eggs that are expelled from a person who has an infection. Eggs hatch, they infect a snail. Inside of the snail, they reproduce asexually and clonally, which is an interesting process. They're released from the snail in another form, which goes in and infects humans when they are in contact with that form of the parasite. And I won't give the technical names there. They're up there. What's important is we have this complicated life cycle. The parasite has the same genes throughout the life cycle, and we have to find that place in the life cycle where we can make cogent observations. And that's something that Dr. Blanton has worked on a great deal, as have others. And I put that red circle around step 1 there, looking at the eggs. Now, what's important to realize when we look at those eggs is we can look at which genes are in a sample of eggs from a person, but we can't look at the individual parasites. So it's essentially, in human genetic analysis, sometimes we do pooled DNA analysis where we'll pool the DNA from, say, people with the disease cases versus controls or something like that as the initial screening step. So we're really, as human geneticists, well equipped conceptually to deal with data like these, but it's important to know what our sampling units really are. Now, the genome of this parasite is quite interesting, at least to me. It has 8 chromosome pairs, which is not so big or so many. But what astounds me is it's a very small genome. It's about 8% the size of the human genome. That would make it less than 1% the size of an amphibian genome. Still quite a bit larger than a bacterium, but it has a small number of genes, and that's because it's a very efficient parasite. As it adapted to its host, it said, I'm not going to make all these gene products that the host is just making for me, and then eventually just discarded those genes and became very efficient in its work. Even with this small genome size, and this is an important feature of it, is there are thousands of variations that can be used in DNA profiling, and that is what is going to allow us to track infections. What type of genetic markers do we use? We use a type of genetic marker called a short tandem repeat. It's also called a microsatellite. Those are basically synonyms. In other words, 2 words that mean the same thing. And I like short tandem repeat a little bit better because it's somewhat more transparent to people who aren't already familiar with them. But the idea is this: if we look, if we look at there we go. If we look at a chromosome at a particular location on that chromosome called its locus, what we find is we have repeats of nucleotides. So in this case, it's a hypothetical example. It would go TGA, 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 TGA. And we call that a trinucleotide repeat. Now, when we look at different chromosomes, what we find is they have different numbers of repeats in there. And just due to the molecular mechanisms of replicating DNA and having repeats, it's not very accurate. And so you have many mutations in these and a lot of variation in populations. We're actually all familiar with these sorts of genetic variations if we watch television, because all of our crime shows, procedurals, they talk about the CODIS loci that they use to match criminal samples to criminals, basically. And they use human microsatellites or short tandem repeats for that. So it's pretty standard. And the thing to remember is that actually, if you look at a genotype of an individual where you would have 2 copies of any particular chromosome and those locations on the chromosome, 1 copy, what the person may have inherited. Oh. All right. Well, may have inherited a long one from their father, a short one from their mother, and their chromosomes are actually slightly different sizes. So the chromosomes vary in the number of repeats that they harbor, and we define an allele in analogy to our classical genes as the number of repeats that run in the allele. We usually don't count the number of repeats directly, but we just measure how long the allele is, and that gives us a good idea about how many repeats are there. In the study I'll talk about, we've looked at 14 of these short tandem repeat loci, which I believe is the same number that the FBI and the CODIS people in the United States use for matching criminals to their evidence. When we look at the 14 loci, there are actually 142 different alleles that are in there. So it's what we would call a hypervariable source of DNA. We measure the frequency of each fragment size in the pooled DNA samples from eggs released by an infected individual. And that's why I like mathematical analysis, because I don't look at the feces that they're extracted from. In my other work, I don't look at the blood, at least not while it's still red, or urine or anything like that. It's very clean if you do data analysis. And thus, we look at measurements at what I will explain is the infrapopulation level. And I'll get there in just a second. Okay, but before I get to infrapopulations, I'll just say that we're looking at samples from 3 communities here. One is called Jenipapo. There were 69 individuals. I should mention that much more data were collected on these people, but we've only used those samples where we have complete DNA profiles for everybody without missing data, and that is advantageous. There's a neighbor of Jenipapo called Volta do Rio. We have a smaller sample size. It's still fairly large, 24 individuals. And then we have São Bartolomeo, which is a community here in Salvador, which I was fortunate to visit the other day. That was very gratifying to actually see one of the locations, the people who collect it, to meet all of my collaborators. It's been a wonderful trip here this week. Our total sample that we're talking about is 254 individuals there. I won't go through all of the methods of data collection because you guys did it and you know it much better than I do. All right, this is a map of the communities. Obviously, this is where we are right now, and you can see that Volta do Rio and Jenipapo are, I think, what, 150 kilometers away or something on that order. There's another distinction. 300. Oh, 350. Okay. Substantial. So 350 kilometers, 200 miles in that range. In other words, those worms aren't walking over there by themselves. And that's what's the important thing. They're not traveling from these, they're not traveling from these rural communities to this urban community on their own. They have to be carried by people. And that's what's important to us. So this is I should just mention, this is a slide that I extracted from a presentation that Ron had made before. So thanks. Okay, now when we look at the genetical structure of the worm populations, then what we find is it's at a multiple of different levels. One level is the individual worm at a particular stage of its life cycle, which we don't really ever see. There are labs that are pulling individual worms out of people, or finding ways to isolate single individuals. But if you were to do that, you wouldn't get an adequate sample size to do the types of analyses that we want to do. The next level is we can look at mated pairs of worms. And this is one of the things I find somewhat amusing about these worms is they actually pair up in a monogamous type of relationship that, I don't know, the Pope would approve of or something like that. But we have mated pairs. And then we have this unit that I've alluded to already, which is the infrapopulation. And what the infrapopulation would be all of the mated pairs inside of an infected person that are depositing their eggs into the vessels and tissues of that affected person. It's hard to say how many there are. Some of the literature sources I looked at said a typical infection might have a median of about 50 mated pairs in there, which is a fairly large number. But it can be small. Obviously, it starts with one mated pair. It can go up into the many hundreds and have very severe infections as well. So these are averages, and we all know averages are only a first step in describing. Above the infrapopulation level, we have what are called component populations, and these are the collection of infrapopulations in a given geographic locality. And then finally, we look at what would be considered a total population, which would be all of the different people and all of the localities that we would have. To sort of illustrate this, I've used dots to represent our worms, and we have females and males. I guess the males are bigger. I've got a picture of that in there, so I didn't show it. And they pair up, so we get the mated pairs. They find their ways into individuals. So we have the infrapopulations. The infrapopulations are nested inside of the component populations, and then finally, they're all together in a total population there. So this is very similar to the way population geneticists have organized their conceptions of diploid populations since the beginning of the 20th century, except that there's this level of infrapopulation, which is not a population in the sense of a persistent deme or having individuals mating with each other over multiple generations. They just spread out at the end of every generation. All right, so I talked about different goals of the study, and I identified three major goals. And now what I would like to do is just talk about this first goal here. Do the Mansonii parasites form genetic clusters that coincide with the communities of their hosts? And one of the things we've looked at from different perspectives that we found is for a couple of these microsatellite short tandem repeat fragment sizes, you will see a high frequency in only one of the communities there. So in this case, we have a genetic locus that's SMMS21, which is just a catalog number, but it's an accepted one. And we find that there are fragment sizes in steps of 3, like I showed you before, but one of them, the 177, is only found in San Bartolomeo. And basically what that would mean is if you examined a person and you found that, they would likely be from San Bartolomeo. We also see that there are a couple of them at a different one of these loci that tag the Volta do Rio population there as well. Unfortunately, or as it turns out at least, there are no definitive tags for the Genipapa population. And so we don't have a lot of specificity for Genipapa. Now, the next step of analysis is not quite so visual. It's where we jump into statistics, and I've decided to use a set of statistics that were developed by the famous population geneticist Masatoshi Nei. And Nei talks about gene identities, and we can look at it at different levels. So if we're looking at a group X, which would be Genepapo or Volta del Rio or something like that, what we find is if we took one copy of the locus and then took another copy of the locus, are they the same? Are they identical? And that's gene identity. And if you think about that, if we were talking about people and we went to one of the short tandem repeat loci, say in my genome, the question would be, does the allele that I received from my mother match the one that I received from my father? And that would make me a homozygote at that locus. But we're looking at these just at the individual profile level and not at the genotypes. So Dr. Nei decided that it would be better to call them identities instead of homozygosity or heterozygosity. The next thing that we want to look at is the gene identity between two different groups. And this idea would be, I would take one copy of a chromosome from one group, and I take one copy of a chromosome from another group, and talk about whether they were the same allele or not. And you can think about this in very simple genetic terms. This would be saying, if a parent, say a woman from one group, donated an egg that was matched with a sperm from a man from another group, would they be the same allele or would they be different? And our intuition tells us that they're more likely to be the same if they're from the same group than if they're from different groups. And now we can define something called a genetic distance between those two groups by saying, what's the difference? How much higher is the probability of identity if they both come from the same group versus coming from two different groups? So this is the only equation that I'm going to show today. There's actually a lot of math behind it. You could make it look more impressive than this, or depending on whether you're impressed or depressed by equations, they could get much more complicated. But I think that I like the idea of trying to explain it in terms of general terms. You know, what's the probability of being a homozygote if your parents come from the same group versus the probability of being a homozygote if your parents come from different groups? And then we might imagine that if the different groups are very far apart, it's going to be even lower chance of being homozygous. Now, what is this? This is a matrix of the gene identities for our population that we're studying. And at least it's a representation of the matrix. Because what we have is, as I recall, we had 254 individuals. So it's 254 by 254, which is roughly 65,000 different little, I call them pixels, but they would be more likely matrix cell. Look at the gene identity in every one of them and then shade them in, color them in. Yellow is low, plum, I don't know what the color, purple is high. And you can see that there's a lot of differences. It's a little bit hard to see, but there's a diagonal going up here. That would be all the gene identities within. They tend to be higher. And then we can also see, for example, that Genepapo and Volta del Rio have more of the purple color. And then when you go off into the region of San Bartolomeo, there's more of the yellow color. And I'll explain some of the other complexities of this as we go along. Another technique that we use for trying to simplify those matrices of gene identities into something that is visible. Obviously, if you put it, you can put it all into an Excel spreadsheet, but you're never going to be able to find patterns because your screen's not big enough. So I kind of like that heat map sort of representation. But we can also use something called principal coordinates analysis, or principal components analysis would be another name for what we're doing. And what this method does is it translates those pairwise distances into a space of Cartesian coordinates that would be simpler. It allows a simple visualization of patterns in the distance matrix, but there is some loss of information there. So whenever you simplify things, you're always losing information, and you have to balance when you have made the right amount of loss of information relative to the amount of signal that you've pulled out. Usually the first 2 or 3 coordinates reveal the main features of large data sets. So what happens when we look at our 3 populations here? Now what we see, I've shaded the Bartolomeo in darker blue, Jenipapo in the lighter sky blue, and in this red-brown color, the Volta do Rio. And this is the first principal coordinate, and it actually captures about 60% of all the variation in the sample. And it separates the 3 communities pretty well. There's some overlap in here where you can see that a few of the voltas kind of come into the Jenipapo space, but they're both very well separated from the Bartolomeo. Now, when we look at the second axis, which accounts for about 11% of the variation, then what we see is it doesn't separate. If we project onto here, those 3 communities would just superimpose on each other. And what this is finding then is that it's separating out members of the same community from each other. If we go to the first and the third coordinates, then we get complete separation of the three communities there. So it's a good representation telling us basically relative to our first question that yes, these communities do form separate genetic clusters that correspond to the community. Now, I should also just mention that this is not a usual pattern to see. Usually what we would see is components 1 and 2 would separate out the communities, not 1 and 3 as we have here. And that if you have something going on within communities, these would come out on separate components instead of the same component. And there was a lot of theory about this that was developed by the human geneticist Luca Cavalli-Sforza that people may have heard of, and his colleagues a ways back. But we do have that community separation. That's good. And that brings us into the next question, is do the schistosome Mansoni parasites form genetic clusters at the sub-community level? Like, are there meaningful differences clustering of parasite infrapopulations within Jenipapo or within Volta do Rio or Bartolomeo? So I've addressed this using 2 different approaches. Once again, each one involving some data loss, but perhaps pulling signal to the foreground. And I computed what's called an unrooted neighbor joining tree from the genetic distance matrix. This is a method that was also developed by the Japanese geneticist Masatoshi Ne. And it is a clustering method that starts out, it just leaves all of the samples individually and starts linking them together. So you link 2 together, then you link a third one into that until finally they all come together into a tree. By contrast, I used a standard method from statistics called k-means cluster analysis, and it takes the opposite approach. It says, if I consider them all one unit and start breaking them up, what do we see? And hopefully the 2 methods, whether you go forward and link them together or backwards and tear them apart, are going to lead you to the same conclusions. So this is our neighbor joining tree, and it's quite satisfying. Here we have the Bartolomeos, we have the Volta do Rios, the Jenipapos. One of the things that's kind of interesting is there's a little Jenipapo blip right there, and it looks like the Volta do Rio, came out of the beginning of that cluster in Janapapo. And they're only a few kilometers apart. We've been talking about it, but it's not a lot relative to what we can look at. Now, when we ran the k-means cluster analysis, we ran 3 clusters and we got our 3 groups back. So that was step 1. But when we ran 4 clusters, what it did was it split Bartolomeo into 2 groups. And there was this subgroup within Bartolomeo. And when we ran it with 5, it kept that cluster there, but it added a subcluster within the Janapapo group there. And then the question is when you get the cluster analysis results, they're just all a statistical list there. And I thought, well, I'll just shade the tree and see how they turn out there. And then similarly, what I wanted to do was go on from there and say, are there genetic relationships between the parasite groups in geographically distant communities? And so I went back to the principal coordinates. And here, what you see are coordinates 1 and 2. This is the same principal coordinate analysis I presented earlier, but now I've shaded the points according to the clusters that they came from. And what we find is coordinate 2 that I said seemed to be a little bit out of line from what our theoretical expectations would be if they were purely tree-like, then is picking out the subclusters from the 2 groups. But once again, this is a very unusual pattern because coordinate 2 is saying there's something similar about this group with that group. And those, as we've just heard, are, was it 300 kilometers apart or 300 kilometers apart? And we don't really see very much of it, maybe 1 or 2 in Volta do Rio, which is right close to Janapapo there. So I've pretty much explained this slide already, that in principle, each branch on a tree should have its own principal coordinate. And yet we see that the subclusters 4 and 5 go together on coordinate 2. And that's something that we would not have seen if we just calculated the tree. It's something we would not have seen if we just calculated the principal coordinates. It was using all of these different methods in conjunction with each other, but critically thinking about what was coming out of it. So now the question becomes, what happens if we try to visualize all of the data together? And we go back to our heat map here. And this time, I ordered the populations, Volta do Rio, like it was before, Janapapo, but I pulled all that subcluster together and put it at the end. Then I pulled all the subcluster from Bartolomeo and put that next to them and put the rest of Bartolomeo out there. And voila, we get this area here that we can pick out of the entire 65,000 points when we look at it. Now, there hasn't been I haven't done a statistical test to see whether that would be statistically significant, but I'm feeling like it's visually compelling enough that I'm willing to accept it with confidence. And so, and there isn't an available statistical test, otherwise I would have. So this is a very exciting finding, because what this is telling us is there is a group in San Bartolomeo that has genetic similarity and relationship with this group in Janapapo, and it is making this what geneticists and Cavalli Sforza would call a reticulate pattern. So reticulated patterns is a fun word in English, but it just basically means squares. It's like the reticulated python that people keep as pets. That snake has that pattern. So this was a truly surprising finding to me. But now what I would like to just do to finish up looking at this is ask the question, what kind of process of movement of these worms, and of course, the worms don't move on their own. We're really talking about movement of people here, but we're observing the worms and the movement of people through the movement of the worms would create that pattern. And one type of model we would consider would be one-way, what we call one-way gene flow, which would just mean people from, in the first case, San Bartolomeo would go over to Jenipapo and establish the infections there. The one-way gene flow could go from Jenipapo back to Bartolomeo. You go in the other direction. I'm sure that people are immediately thinking, oh, it could go both ways too. And we thought about that, but I didn't want to go through all the possible submodels. And I think you'll see that this will take care of it. Another possibility would be that there was migration into both of those sources, both of those communities from an outside source. And I want to look at those patterns in the evidence that we have and try to decide between these different possibilities. So this gets us down to our third question: are there genetic relationships, and how we're going to model that? And if we look at the one-way gene flow idea, were there people who carried worms from San Bartolomeo to Jenipapo? Our data actually rejects that idea. How does it reject that idea? One way that it rejects the idea is, recall that we had this population group-specific marker of Bartolomeo. It's extremely common. And if you took 20 people from Bartolomeo and brought them over to Jenipapo, there's almost 100% chance that you would see that allele also in Jenipapo, and it would no longer be a marker of Bartolomeo. So we can reject that. There's a subtler way, is that when we look at this square here, what we find is this group in Bartolomeo are really highly related to each other. This group in Jenipapo are highly related to each other. And then there's relationships across in those portions of that square. Now, if we just had one-way movement of a random sample from Bartolomeo into Jenipapo, then we would get these areas here and perhaps this area right there. But there would be nothing to make them similar to each other, any more similar to each other than the people, any pair of people in, or infrapopulations, I should say, in Bartolomeo. Going the other way, recall we didn't have a population-specific marker for Jenipapo, but we can still revert back to this pattern here. And when we revert back to this pattern here, we would not see that cluster stick out, because if it was just a random sample of people from Jenipapo, you wouldn't see that blip right there. You'd get something that looked kind of like a whole square in there. So we reject that one. And then finally, what we find, and this is not so visually apparent because it's consistency, but if we have this idea that there is some outside source contributing to both populations, then it's not going to take away those group-specific alleles. They're going to remain group-specific, as we saw in Bartolomeo. And it would create those blocks that you see that would not quite be the same in the situation of the one-way gene flows. So the model that I would say that's most consistent with the data is this model 3 here that would postulate that there's some outside source of migration into it. Now, when we think about it, once again, we are not talking about migration of people per se, we're talking about migration of worms. And so when we see an outside source contributing to these, it could be that there is some community out there that has different characteristics that has sent settlers into both of the human communities. But it could also be something much simpler like that than that. It could be that these people go to a common location, they don't live in that location, they just somehow pick up the infection when they're visiting a different community and carry it back to their own. So once again, we're just looking at the movement of the worms and not necessarily the people themselves. Okay, so overall, and this is taking everything I've talked about so far, I would say that when we look at the genetic structure of the schistosome mansoni, we have a lot of local isolation. Otherwise, we wouldn't have distinctive communities and population-specific markers, but that's occasionally broken by long-range migrations in there. And so that's an entirely plausible population structure. It would be, if there's an outside source, it would be very gratifying to be able to identify that source. It is surprising to see genetic relationships between subgroups in widely separated communities. And that's a little bit frightening to me if you're thinking about controlling infections, because that suggests that there are very distant sources that can establish new infections and contribute to existing infections in communities, and that any permanent eradication of the schistosomes will require both the local efforts, but also identification of regional reservoirs of the infections. So it's perhaps to the epidemiologists not surprising whatsoever that this is a very complex situation, because many years ago now, the World Health Organization wanted to eradicate schistosomiasis altogether, and that hasn't happened yet because it's a complicated situation. And then finally, I'm just going to end by thanking my colleagues here. So obviously it was Ron Blanton who talked to my colleague and friend at the University of Michigan and put us, Mike Benke and Mike put us in contact with each other, which has turned out to be really interesting to all of us. Sarah Taylor was a honors student who worked with me on this project and ran a lot of the analyses. She's a graduate student at the University of Michigan now. And then, of course, the final lineup of authors here needs no introduction in this audience. The work was, I believe, funded at least in part, if not totality, by a grant to Ron Blanton. And then the last point I'd like to make is that all of the interpretations and errors I will have to take full responsibility for, although I have taken feedback from my colleagues, but still, that's the way it is. All right, thank you so much. I'd be happy to answer any questions that you have.
Thank you very much, Jeff. I think that was extremely interesting. I would like to just add a little bit to your presentation. That all genetic studies, they all look sophisticated and sexy, but they really depend on the context in which they exist. And the context for these studies is the fact that we had a symposium here during the whole week on urban schistosomiasis. And the urbanization of endemic diseases is something that I think is more and more recognized here in Brazil, and is the future of other places like in Africa as well, the urbanization of endemic diseases. One of the questions that we had phrased another way is, well, what is the presence and permanence of infections in an urban area? What are they due to? And generally, we think of migratory patterns, and the complexity of those migratory patterns that Jeff alluded to is perhaps going to the ability or the facility with which we can eradicate those diseases, or eliminate those diseases as a problem of public health. In addition, they have implications for things like antimicrobial resistance or anti-schistosome resistance. Should it arise, are the other implications of these patterns. And although we generally think of a pattern of going from the rural area to the urban area, and that to be a rather simple process. I think what you have shown is that there are many more contributions than we might imagine. At this point, gostaria de abrir a sessão para perguntas para pessoas aqui ou no chat.
I don't really have a question because I'm part of the work, but I would like to say thanks to Jeff. And when I start genetics, you learn a new vocabulary, a large vocabulary. And you was so brilliant that simplified everything to the audience. And again, when you do genetics, you think the guy is a biologist, no, the guy is a mathematician. and you hide all the numbers and draw these graphics that are so clear to everyone. I'd like to thank you for that. And say that I was talking with our friends that the new data you bring to us give us more homework to do. Because we need to identify the source of this possible source of infection in both communities. And we will do this sooner or later.
Well, I'll just make one comment about that, is that I've always amused, I talk to colleagues and they say, if we just had more data, the questions would be answered. In the course of my career, what I found is every time there's more data, there's more questions. And so we never really get ahead of, and maybe even further behind. But it has been a pleasure doing this. And there are a lot of mathematics in population genetics, and I really love them. It helps structure my thinking, but I do them so that I can understand biology. And so that there's really an interplay there, and maybe even the mathematics and the biology are the two different sides of my brain or something like that, because I love them both. I like to have them work together, but they really, it's not one or the other here. So, yeah.
Thank you, Professor. Congratulations for your presentations that are very. I would like to open to Professor Eliana Azevedo to talk to us and to make questions and observations. Bom dia, professora, pode ficar à vontade.
Tem que abrir o microfone, Rodrigo. Ela.
Professora, a senhora pode abrir o microfone, porque está fechado. Professora Eliane Azevedo. Tá.
Não, tá ali.
Pronto, pode falar.
Yes, I would like to thank Dr. Long for very
Só um
interesting.
minutinho, professora, que está baixinho aqui. A gente vai consertar. Pode falar, obrigada.
Thanks to Dr. Long for a very interesting talk. Excuse my English, I am 88 years old and my English is as old as myself. I have a question. late, many years ago, some studies in Brazil showed that I don't know what happened. Can you hear me?
Yes.
Yeah.
My everything became blank in my notebook. I cannot see anything. But if you are hearing me, if I can hear you, I will continue.
Nós estamos vendo bem e ouvindo também. Você pode continuar.
Okay. I feel very happy to see analysis that have been developed for human population structure being applied to schistosomal populations. And my first question is, when Dr. Longren do the statistical analysis, if it's done under the assumption that the gene distribution is under the Hardy-Weinberg equilibrium, considering that the schistosomal population has male and female. The other question related to this one is if gene identity is the same as we call homozygosity. Finally, I noticed that there is no reference to the racial background of the donor of the fetus. And many years ago, Dr. Bina and myself, we published on the human biology a long study made in Caatinga do Moura in Bahia, showing that the Afro-descendant, they do not develop the severe form of schistosomiasis. This was very clear by following those patients for many years. When they are black, the infection remains in the intestinal form. But if they are white, they develop to the severe form involving liver, a spleen, and the portal hypertension. I wonder if, as I know, the assumption was because since the Negro came from Africa, where the disease was endemic, they had developed some immunological resistance to the severe form of the disease. However, we never saw any data proving that. I wonder if some relationship could be established between the genetic constitution of the patients, if Afro-descendant or if white, related to the type of genes identified in the infected organism, the schistosomes. I don't know if I made myself clear. Thank you. I'm very happy for listening to Dr. Long. It was very, very interesting working. Congratulations.
Well, thank you. In relation to your three questions, we do not need to assume Hardy-Weinberg equilibrium for these analyses. And that's why Dr. Ney called it gene identity rather than homozygosity. And if there is random mating in the populations, then they will be homozygosity and heterozygosity. I feel that random mating on a local level is very robust, and that we see that a lot. Now that we have the Human Genome Project, we can test it on tens of thousands of loci altogether, and I find that there's good correspondence to it, but we don't need to make that assumption. So that's just as well. And then your final point about the population affinities of the people who are carrying these worms, and whether there's differences in susceptibility or disease course on people of different ancestry backgrounds, I think is a very interesting one. And that isn't one that we've been able to address yet, but it might be possible, and it would be an ideal step to look at in the future investigations.
I would make one comment. We are studying parasites, but the one thing that I would comment about infectious diseases in general is that we almost are not studying the pathogen as much as we're studying the host. That is, all infectious diseases say an awful lot about us more than they do about whether the pathogen we're looking at. If you look at the distribution of COVID infections, I don't explain it, but the fact that there were more infections and more disease in the United States than in China says something about the way that the U.S. is constructed socially. That there were very few infections in Africa, or I shouldn't say few, there were lower in many parts of Africa, not in South Africa, some other places. Says a lot about the way their society is structured and put together. And when we trace these patterns in schistosomes, we are tracing actually patterns in terms of humans. One pattern that we do notice is if you look at Lopodolio and Biddy Popple that are on the road 5 kilometers apart. And they have relatives back and forth and they move back and forth. But the important part is that probably they are not having their bowel movements in somebody else's backyard. That the genes stay home because that's the human pattern. And later on, we'll be looking at in urban areas. What we expect to find as well is that even though they're even closer to urban areas, we may well see that they are limited, restricted to certain areas simply because people do not have those habits of moving between places and also having their bowel movements in somebody else's backyard. Again, looking at all this genetics of the parasite, I think says a lot about human populations.
Temos uma pergunta da história.
Hi, I'm Lucio Robo. I guess you know me, but
We've met.
I don't have many questions as we were saying, where I'm part of the work, but now that you're presenting, I started wondering, could we use something similar to the molecular clock to relate these populations between them? I mean, as you said, it doesn't look like the migration went from São Bartolomeu to Genipapo because of that allele. And I was wondering, I never even considered before analyzing this way, but now that you mentioned, could we use something like the molecular clock, or?
I think the molecular clock's a little bit like the human memory. There's sweet spots where it works. The short term isn't always so good, and the long term isn't always so good. And this may be too short term, say, for evaluating Genipapo and say Rio Volta do Rio. Right, Ron is interested in expanding studies to Africa, and there it might be a very useful approach to do it. The migration patterns also make the molecular clocks a little bit difficult to apply, because usually they assume that there's there's complete isolation. I wonder about these long-range migrations. I think that, you know, Ron brings up this point that people don't defecate in other people's backyards. But sometimes I wonder if the further you are from home, the more likely you are to do that. If you're close to home, you're not going to do that whatsoever. But if you're traveling a long way and maybe you've become ill in the process and you have no home to go to, that may establish some of these long-term infections. But I don't, you know, that's wondering about things. So
I actually agree with you. And I say that it's epidemiology. We're talking about, for the most part,
right.
statistically, because what you seem to show, and I agree with, is that these were established by migrations initially.
Yeah.
The question is how frequent is that, and you don't often travel.
Right. That's, I mean, and that's a good, I mean, that's a good line for future research, of course. On this.
Yeah, I would like to stress about one thing that I always, I'm curious. When you do genetics in a setting like the Jenny Papa and Baltimore Hill, it's a very small town.
Yeah.
We don't say she's to alt infection, but the alt correlation is quite reasonable that happens. Because the output is in the river, they got infected in the river, so the genetic pool is limited to this particular setting. But in an urban area, we have an example next door in Batifacho. You have Cercaria in the water, a lot of Cercaria in the water. Every month they collect the snails shedding Cercaria, but the prevalence is very, very low. So not many people get infected in this place. So it means that the schist is coming from the output of someone else from another neighborhood that's coming from the sewage, contaminate the environment. And you may have infection that come from somewhere you don't even know in the room.
So I think that that's, you know, those are excellent thoughts. And just to take them a little bit away, that's why I feel like I've gained so much by being here this week and listening to all the health department people talk and looking at the water management, looking at the kinds of pipes they use, looking at the color of the water and the photographs. It's just helping me so much more to think about it, because other than that, I'm just sitting there with these matrices of numbers and trying to find patterns in them that mean something to people. But yes, I think the points you bring up are really excellent points, and you know, hope I eventually learn more about those matters.
Thank you. We have a question from the chat. From Dr. Leonardo Farias, researcher here. Thanks, Jeffrey, for the great talk. Are you planning to analyze on these samples potential mutations in the ion channel recently associated with the mechanism of resistance to PZQ?
That would be a great thing to do. I don't have any plans. It's not on my computer right at the moment. But if the data were to come my way, I would be happy to be part of that and assist with it. So.
We are not planning to embark because everybody has their own biases, and scientists do as well. And my bias, which I think is based on evidence, but anyway, my bias is that protoconal resistance is not a problem. I've not really seen it truly, even in Africa, the pattern that you see with resistance to malaria, anti-malaria, is that it starts in one place and then spreads to somewhere else. We've never seen that. It's just biases. And when the gene has been identified, and finally the gene has been identified, that I feel confident is responsible for quasi-quantum resistance that occurs in the laboratory. And you can make resistance in the laboratory at the drop of a hat. We've not seen that in nature. And when they've gone to look for those genes in nature, they've actually not found those mutations in natural population, which probably suggests that it is not a mutation that is viable outside of the laboratory. Yet, quasi-quantum resistance will probably exist at some point, but not so far. We have looked at resistance in our studies, both in the rural areas and the urban areas, and they're so far in a decade, and with yearly treatments in some places, we've seen no evidence of resistance really, although Trevor said 100%. In kind of it is, but usually it's more like 80 or 90%, and usually if you do treat that 20%, can be taken care of. So we've not seen resistance, anything that looks like resistance to quantum in Brazil and certainly in our studies. The first year we had 10% of people who were still positive after we treated them. There were mostly people who were previously heavily infected. But in subsequent years, over again a decade, we would have 1 or 2 people who were still positive, and those people would be treated. We really haven't seen anything that looks like resistance. So there are no plans to do that so far, in part because, at least in nature, there's not much to study in Brazil for resistance.
I would say I'm very like-minded with the way you're thinking, but I might also add, and maybe this is my bias, but I think it's much easier for something like a virus or a bacterium or even a single-cell protozoa like malaria to develop resistance than it is for something with, I talked about what a small genome it is, but it is still much bigger than a bacterium or probably bigger than malaria. I don't know about that, but I think that just because of the way eukaryotes work with mutation rates, with reproduction and things like that, the situation is not as ideal to develop drug resistance. But like I said, that's my bias. It's just shooting from the hip. So
Thank you.
Yeah.
Now, Dr. Vickemeyer, you also make a question.
I don't have a good question. First, I want to congratulate that. Very nice talk. Very nice talk. Very nice. Very well. I will share some from the result that got. I think that it's a lot of pressure for us in order to refine our epidemiological analysis and get more social demographic data, just to explain what are in common between people here, the schistosome in my population here, what of the Rio or the Genipapo. Historically, basically the Portuguese and that went to Africa, and the African that was fought here by the Portuguese, they came to Salvador. So my feel that schistosome was in Salvador, and then a lot of movement here. The African people just they move out of Salvador just to surround the bay, then they go to the interior of the state. That's one point. That means probably the world were produced in Salvador, like Científica showed in 1908, and then moved to the interior. When the people just try to escape from here as a slave and went to their countryside. On the other hand, the Genipapo and Volta do Rio, I don't know, I probably did not mention it to you. We had a train line that made the connection between Salvador, it's not really Salvador, but the other side, to that area. I know that what one say that somebody wants to food, but probably someone wants to Volta do Rio, stop there, do there, and go over there. That's one thing. So we need to refine this. The other situation that we're facing now is the opposite way. The very strong demographic transition, where people come from the countryside, the rural area, to the city here. And I always try to remember, I used to work in a place, a city called Itaquara, where it's called Rome. And 2 or 3 families that everybody were infected, high level of infection. They came to Salvador to live very close where is São Bartolomeu today. They went to Valéria, but they moved to probably Pirajá. So this, I see, put a lot of pressure on us to refine our analysis and to get more information, more data, and find the analysis at the epidemiological level to see if what are seen in common as the parasite in the population could be something related to this people movement. But congratulations for your talk. That's a really nice talk.
Well, thank you. And I just, have to say that I think it would be very exciting to expand these data collections and see if we could learn more about all of these different possibilities.
Very well. I'm going to thank Jeff and all our participants, everyone that participated this week in the symposium on as well as the institutions of civil construction, sanitation, and public health that participated this week. And again, thank you all for coming.
Thank you, everybody.
Thank you.
Thank you very much, Doctor.